Have you ever had a parts list of a kit you needed to build? How about the components of one of the simplest cells on Earth? It's extremely complicated and the list is jam-packed. It's so crowded, the parts have been described like this. The interior of the E. coli cell makes Hong Kong look like Nebraska. Coming up next, the Dictionary of Life with Dr. Paul Nelson. Hello, my friends. My name's Donn Chapman, and you're watching Origins, and we're delighted that you are. Thanks for joining us. You know, Origins is a place where we take the evidence of science and use it to validate the truth of creation. We do that by interviewing some great guests, and we have a terrific guest with us today. Dr. Paul Nelson's here. Dr. Nelson, it's so good to have you.
Thanks for having me.
Dr. Nelson has his PhD from the University of Chicago, which is kind of a historic place when we're talking about creation and evolution and all that, isn't it?
It is. In fact, in 1959, at the 100-year anniversary of Darwin's Origin of Species, they held a huge event with scientists from all over the world. So it's a center for evolutionary research.
And yet you came out of there believing in intelligent design. Actually, you went in there believing in intelligent design.
Yeah, it's a wild story. I guess I'm a bit of a rebel. But I love the school because they gave me a fellowship knowing that I disagreed with Darwin.
And that is a sign of a great place. You go by the title philosopher of biology. I love that.
It's the place where the science of biology meets questions that are classically philosophical, like what counts as a good theory, or how can we test this idea if we don't have data? You know, questions that belong really to the realm of philosophy that intersect with science.
Yeah, that's tremendous. And you're teaching that at Biola University?
Yes,
and
Southern
you're
California.
Also a fellow at the Discovery Institute, which I think is doing some tremendous, exciting things. So thanks for being with us. We're going to talk today about the Dictionary of Life. Now, you know, I think of parts lists. I put things together for my granddaughter, for my wife. And, you know, you put slot A in hole B and put on nut C. Is that what we're going to do today, look at a parts list?
You know what, we are. We're going to look at a really interesting parts list. It's the parts list from one of the simplest cells on Earth. And I want to show you just how complicated it is.
I think this is going to be tremendous. So let's do that.
All right. Here's the cell we're talking about. It's called mycoplasma. It's actually a parasite of human beings. Lives, for instance, in our airway. Very stripped down cell. Because it's bathed in nutrients all the time, it doesn't make any amino acids for itself. So it's got rid of a lot of hardware, but it still needs a lot to function.
Wow, look at that list.
Yeah. If I saw that in an IKEA kit, I'd send it right back to the store.
That's a lot of stuff.
So I know this is going to be hard to read, but I'll show you what's here. These are proteins organized in different functional classes. This should be easier to read. So you've got proteins involved in building the cell envelope. That's the boundary between the cell and the rest of the world. You've got cells involved in energy metabolism, converting the raw materials the cell needs into forms that it can use. There's proteins involved in DNA translation, taking DNA, turning it into protein. DNA transcription, taking DNA and turning it into RNA. Cellular processes like dividing, we'll come back to that. DNA replication, making copies of the permanent information. Transport and binding and so forth. So you can see that these are different classes of functions that...
All those functions have to take place in the most simple cell.
In the most simple cell. And I've only listed 8 here. There are actually many more, including some that we don't even know yet. come back to that. So let's look at some of these. This is a protein that has a very interesting job. You know, when human beings reproduce, we don't, find a nice dark room and put on the candlelight and say, now my midline is right about here, and I'm going to split myself in two, and one Paul Nelson gets to go, to Pittsburgh to make a TV show. The other guy has to cut the grass. Humans have more interesting ways of passing on their genetic information. But for a bacterial cell, finding your midline, the 50% point, which is right in the center, is very important because you've got to get a whole chromosome in one half, a whole chromosome in the other, and split down the middle. The job of this protein is to locate that midline. That's going to be the place where the bacterial cell will divide. Now, keep that protein shape in mind. Think of it as a molecular tool, and let's compare that shape to this one.
Oh my.
Now, this is actually a multi-protein complex that, as its name implies, is chaperoning or supervising a particular process. When proteins are newly formed, many of them, as they come out of the molecular machine that makes proteins, the ribosome, they're very long and sticky. And the interior of cells, bacterial cells, is incredibly crowded. Jim Shapiro at the University of Chicago told me a few years ago, he said, the interior of an E. coli cell makes Hong Kong look like Nebraska. He said, it's really jammed with stuff. So to fold properly, the chaperone provides in that central space a kind of nursery for baby proteins. So they're here folding, and then what the chaperone does is it releases them into the cell once they're properly folded to perform their function. Now you can see that this shape is different than the preceding one because it's performing a different job. Okay, now let's look at another one. This particular protein, again, has a different shape because its role is actually in the big machine, the ribosome, that's making proteins. Now, we could go through that whole list of parts, and each of these proteins would have a different form. Just the way if you went home to your toolbox and popped the lid and looked inside.
So these are the pictures of all those things listed on that chart.
Well, 3 of them anyway.
3 of them, but,
yeah,
but it gives you the idea that every one of those has a picture that would go with it.
Everyone has a picture that would go with it. You can actually go to the protein data bank online and download these diagrams, and it's amazing the diversity of form that they have. All right, so I look at something like this and I think, is all of that really necessary? How many of those parts does this cell really need to survive? Well, Mycoplasma genitalium was one of the first species on Earth to have all of its DNA mapped. When that was done, the biologists who did the project asked that question. They said, we could probably get rid of a lot of this and still have the cells alive and cultured. So they did a very interesting experiment to try to answer that question. How much can we take away and still have a functioning cell? So here's the first time this experiment was done. And I remember reading this paper when it came out, and it gave me goosebumps. And I'll tell you why in just a moment. But this is the first time it was done in 1999. Then it was done again and published in 2006 to try to fix some details, make it a better experiment than the time they did it previously. But here's what they did. They used a piece of DNA called a transposon, and this is blown up so you can read the name, that integrates itself randomly into other DNA. So here's the circular chromosome, or a cartoon of it, of mycoplasma, and what this transposon is going to do is come crashing in to genes. So now we have, let's say this is an essential piece of DNA that codes for a protein in the ribosome, the machine that makes proteins. The transposon randomly integrates itself, disrupts this gene, and if it is essential, you'll get a signal. Those cells will die. So it's like taking your car and just disconnecting cables in the engine randomly. Some of them, maybe your windshield wiper won't work, but you can still drive the car.
Okay.
You pull another one, you take down the whole vehicle. So this transposon comes in, knocks out an essential gene, and the cells die. Well, think about it this way. If you do that experiment enough, you can figure out where the essential genes are on that circular chromosome. So in this case, at least one of these transposon insertions has hit an essential gene. The cells die. In this case, same thing. At least one of them has hit a gene you couldn't do without, cells die. But in sometimes, when the experiment is done, the transposons integrate, but they never hit an essential gene. Those cells will grow. The fact that they grow tells you that, these points on that chromosome
Are not essential.
are not essential.
Okay.
Well, you can map and figure out where the essential hardware is, working your way backwards from these cell populations that are still alive. Now, here's what they found that gave me goosebumps. This is in the 1999 experiment. Think about that parts list. When they did this analysis, they found that 382 of the 482 protein coding genes in that bacterial species are essential, including 110 genes of unknown function.
Wow.
Nearly a third of that essential hardware was doing jobs for that cell, and they didn't know what those...
We still have a lot to learn, don't we?
I tell you what, we have a lot to learn. And what's striking about this is not what you would get from a biochemistry textbook.
No.
That tells you we pretty much understand the biochemistry
In fact,
of
we
life.
could throw in this word. There isn't a lot of junk in there. It's all useful stuff.
Certainly for bacteria, everything is essential. Now, in 1999, when they first discovered this, they said the presence of so many genes of unknown function in the essential hardware suggests that we don't really understand basic biochemistry yet. Now, that's one result, but there's a significant and I think even deeper result for evolution. More recently, this was published last year, a group at Stanford looked at a different bacterium called Colobacter. It's found in fresh water. And they did the same kind of experiment, knocking out genes to see what the result would be. What they found was 90 segments of unknown function. Lucy Shapiro at Stanford was the senior investigator on this project, and she found the results very surprising. In fact, here's how she put it in an interview. She said there were many surprises in the analysis of the essential regions of Colobacter's genome. That's the bacteria they were looking at. For instance, we found 90 essential DNA segments where we have no idea what they do. That's remarkable. These may provide clues to lead us to new and completely unknown bacterial functions.
You know, Dr. Nelson, it seems to me that these folks continue to be surprised that it's even more complex than they think it is, and these are not novices just finding out cells are complex, but even the people in the field. It keeps getting more complicated and more complicated as they look more deeply into it, doesn't it?
It's remarkable. And this has been enabled by technologies which allow biologists to look at all the DNA of an organism, of a species, not just a few of its genes. But here's the real kicker, or the real challenge, I think, for evolution. So on the left here, we see Colobacter's proteins. And you'll notice that here is a significant fraction of essential hardware that belongs just to that species. Now, you can compare Colobacter to E. coli, which, according to evolution, is one of its relatives in the bacterial tree of life. E. coli, which are the bacteria in our large intestine, has its own essential hardware that it doesn't share with its relative. Now, this should be a real puzzle for any evolutionary biologist.
Because they're supposed to have a common ancestor, but there's over 350 parts that are totally different when you add the two together.
That's right. If they shared a common ancestor, they would have inherited the same equipment, essential equipment, from that ancestor. Yet it's clear that they're doing things in a significantly different way.
Is it too strong to say that they couldn't have the same ancestor?
I think you could still entertain that hypothesis, but at some point, as you go back to the common ancestor and look at the branching, you're going to have to integrate a lot of unique equipment here, and it's not just, secondary bells and whistles. It's right there.
It's the essential to run it, yeah.
Yeah, right there in the basic hardware. So here's a metaphor, I think, that may help people understand this. Let's say you go to an auto show, and, hoods are up, you can look at the engines. First car you examine, you see, wow, it's got essential parts A, B, and C. Maybe I expected those. Anyway, I move down the line. Now, C is the same, but D and E are unique for this vehicle. They're simply not present over here. You walk a little farther. B is shared with that car, but then you've got unique parts G and X that weren't present in the other two. And the same pattern holds as you move down the line.
You know, that's a fascinating illustration. I think back to the days, you know, when Henry Ford first made the Model T, he wanted to make every part where if you had a half-inch open-end wrench, you could fix everything in there. A screwdriver and a half-inch open-end wrench. But think today of how complex cars have become.
Yep.
Now, cars are made by people, so we have made them more complex. But when we're looking inside cells, we're looking at what God made a long time ago, and we're finding out it was very complex from the beginning. Just because you have two V8 engines made by two different manufacturers, the essential parts certainly aren't the same.
That's right. In fact, when my wife and I bought our most recent car, the Honda dealer told me, don't even bother looking in the engine, it's not for you. Meaning that's too complicated, don't mess with it.
Yeah. And we're finding that about the simple cell as well, aren't we?
Well, the simple cell is not so simple.
Yeah.
And the amazing finding that I just can't get over is that DNA sequencing technology has enabled us to really begin to map out the genetic diversity of life. And it appears that cells don't always do things the same way, and those differences may be original. They may go back to the original designer.
So they may have a common creator, but they don't share a lot in common. We're going to talk about the implications of that. Don't you go away. We'll be right back.
Today's guest on Origins, Dr. Paul A. Nelson, Ph.D., is a philosopher of biology and evolutionary theory. Dr. Nelson is currently a fellow of the Discovery Institute and adjunct professor in science and religion at Biola University. Paul lectures frequently regarding the intelligent design debate at colleges and universities throughout the U.S. and Europe. He has appeared in several films on intelligent design for Illustro Media. One video he would like to make available is titled Metamorphosis. To order the DVD, call 800-266-7741. To contact Dr. Nelson, write to Biola University, 13800 Biola Avenue, La Mirada, California, 90639.
We are back, and we're glad you stayed with us. So, Dr. Nelson, as we look at this incredible additional knowledge about genetic diversity, what are the implications of that?
Well, if you look at the perspective of an evolutionary biologist about the origin of life, he's going to tell the story beginning with basic chemistry. It's often described as a bottom-up approach to the problem. You begin with non-living chemicals that somehow are able to form something like a self-replicating RNA molecule, and then that becomes captured in a membrane. And if you've got self-replication, you could have something like, you know, a Darwinian process of natural selection begin to operate. The problem is top-down analysis, what we've been talking about before the break, where you look at the hardware required to have a living cell at all, tells you that you need a certain irreducible amount of equipment, molecular players, proteins, the things that do the jobs for you, to exist at all. So if we look at what non-living chemicals can do and compare that to what our top-down analysis is telling us, those two modes of looking at the problem need to intersect. They need to meet in the middle. And the question we have to ask is, do they? And the answer is no. There's a vast complexity gulf that separates the two regions. Non-living chemistry may be powerful, but it's pretty stupid. And it doesn't build you proteins. If you want proteins, you need a cell. That's the message that comes back to us from nature. And I think the reason that people try to build, biologists or evolutionary theorists, try to build cells bottom up is not really, you know, because they're following the evidence, but because they think that's the kind of story they have to tell.
Sure.
And that's a problem.
We have to go from the simple to the complex.
They think they have to go from the simple to the complex. But for instance, take RNA itself. Essential part of every living thing that we know. There's no pre-biological pathway to RNA. If you find RNA on the planet today, either it was made by a cell or it was made by a very clever biochemist. RNA does not form naturally from its parts. In fact, if I were a designer who wanted to leave an elegant signature in every living thing on Earth, I'd use a molecule like RNA that's very unstable and fragile on an early Earth, scenario without a cell to protect it. So our bottom-up story can't even get started. That's a real problem.
This is amazing data, but how widely accepted is it?
The data, that's accepted. I mean, the evidence is what it is. The problem arises when we ask, what is the evidence really telling us? And at that point, you bump into some philosophical obstacles. Science you can think about as a game played by certain rules. And if you've got a rule that's restricting you, the evidence can be clear, but the rule is preventing you from making the natural inference. When I see evidence like this, to me it suggests intelligent design. And we ought to be able to ask that question. The problem is we've got a rule standing in our way. There it is.
The statements of science must invoke only natural things and processes.
Think about that verb must. That's an imperative. That's what you say to your kid when he wants to go to the mall and his homework's not done. He doesn't have a choice. And to drive the point home, the National Academy of Sciences here says only natural things and processes, where the contrast class would be intelligent.
So a designer is sort of ruled out of as an option before we ever look at the evidence.
As a ground rule, it's kept out. That's not healthy for science, because you could have crystal clear evidence of intelligent design, but if you take this rule seriously, it will prevent you from getting at the truth.
So how do we change this?
Well, one of the things I think we need to do is recognize that this wasn't the way that science always functioned. David Hull was a brilliant historian of science at Northwestern University. And in the early 80s, he pointed out that the Darwinian Revolution was as much concerned to change the rules of science as it was to offer a theory about the origin of species. If you go back before Darwin, look at Newton or Kepler or Boyle or Galileo, they had a richer toolkit of explanation, including intelligence, that they could call on.
And how do we get back to that?
I think we need to be bold about saying, look where the evidence is leading, and also take a look at papers like this. Eugene Koonin is a biologist at the NCBI in Washington. He runs a big lab there. And in this paper, which you can download freely off the web, it's an open access paper, he says the problem of the origin of life is so hard to solve that we need to invoke a cosmic lottery. Make the universe big enough that this could happen just by chance, never mind the details. Now, when a scientist comes to that kind of conclusion, I want to say, let's open up the range of options to include design, because this is really not a scientific argument,
no,
but going to a cosmic casino and hoping you get the ball to fall in the right place on the roulette wheel.
That's not my definition of science.
Not mine either.
So we may have built our own little box that keeps us from the truth.
When you have a rule that's standing in the way, you need to move it. I'm old enough to remember the NBA before the three-point shot.
Yeah.
The game is more interesting now with that arc painted on the floor than it was before. But there had to be a point where the NBA said, let's improve the game, let's change the rules.
So it isn't science that is eliminating God, it's scientists that are eliminating God.
I think what it is, if I can follow up on your remark, is a problem not in the evidence itself, but in what we'll let that evidence tell us, in the philosophical foundations.
How close are we to where, as a scientific community, we open the door to say this could have had a designer?
I'd like to believe, and I think there's evidence supporting this, that that point may be arriving very soon.
But sometimes when I hear evolutionists talk, it seems to me that they're basing their whole life on a theory, a 19th century theory that predates the scientific evidence we now are using.
I think there's a lot of truth to what you say. And in my eyes, the real problem is the philosophical foundation of the theory, which says if you're going to do science, you're restricted to a toolkit of just material physical causes. We know that can't be adequate, though. You know, just look around you. The people watching this program, for instance, are watching it on devices that require intelligence for their very existence. And I think intelligence is real. It's part of the universe. It brings about effects that can only be explained by reference to a mind or intelligence. And a healthy science will use all the possible causes at its disposal, not setting some aside, saying we're not going to use those, we don't like the implications. Science needs to move forward, and if we've inherited a bad philosophy of science from the 19th century, as I think is the case right now, we need to get rid of that and try something better.
And friends, you don't have to wait for the scientific community. You can believe the truth today, because remember this, it's God's view that He created you, and that should be your worldview too. Hope to see you again soon here at Origins. And Doctor, it's so good having you with us.
Thanks again.
Bless you.
Thank you for watching this edition of Origins. If you'd like a copy of the PowerPoint information presented today, you can download a PDF file from our website at www.originstv.org. Or for DVD of this series, send a $12 donation to cover shipping and handling and write to Origins Program, number 1301, Cornerstone Television, Wall, Pennsylvania 15148.
Origins is made possible by the faithful prayers and the financial support of you, our Cornerstone Television family.